And that second speaker will be Madeleine Lépine. And Madeleine is a PhD student at INRS, and she was the recipient of the CIAPE evertéa PhD Student Award. And her work focuses on endocrine disrupting chemicals and the development of innovative tools to have in vitro models that can be used to assess the health risks associated with chemical mixtures. And she is going to provide this presentation on developing a new 3D in vitro tool to study the risks associated with exposure to a brominated flame retardant mixture on breast cancer. Madeleine, I look forward to your presentation.
Thank you so much. So hello everybody. Today I'm pleased to present to you in a few minutes a chapter of my doctoral project on the effect of my mixture of brominated flame retardants on breast cancer. So as you know, the advertent exposure of humans to numerous chemicals through divergent routes establishes a mixture context to which humans are constantly exposed to, and more evidence tend to show a correlation between environmental factors and hormone-related cancers, including breast cancer. So my project focuses more specifically on flame retardants, on brominated flame retardants, a family of compounds that were created to delay the spread of flames, but that were found to be ubiquitous in the environment and to have toxic effects. Now, this family includes a large number of subfamilies such as PBDEs and compounds such as HBCDD and several others. Now, in traditional chemical risk assessment, toxicity of a substance was identified, for instance, with the highest administered doses that does not produce a toxicological effect. And this concept was first introduced by Paracelsus with his famous quote, "All things are poison and nothing is without poison. The dosage alone makes it so a thing is not a poison." However, the toxicological profile of a chemical mixture is not simply the sum of its components' effect. It can be altered by synergistic, additive, or antagonistic interaction between substances. And this realization has led to urgent calls for the assessment of chemical mixtures' effects on human health and in the environment. So in my director laboratory, brominated flame retardant mixture were used to evaluate their toxicity on the mammary gland development and breast cancer using rats, dams, and pups exposed orally and in utero, respectively. The results showed alteration of self-junction pathways, hormone receptors, and mammary gland signaling. And in a second experiment where the rats were also exposed to DMBA to induce mammary gland cancer, the results showed in the dams an increase in the breast lesion, in cancer-associated pathway alterations, and in the pups, there were also delays in the development of the induced cancer. Now, these results represent an advance in understanding the effects of these compounds in a mixture form. However, it took more than 15 years of work to publish all of these results. Conversely, since the development of molecules is very rapid, the scientific community has recognized an urgent need to develop faster and less expensive tools than the current animal-based risk assessment models. Among these, new approach methodologies such as NAMs, are generating increasing interest. This brings us to my project, which aims to develop new risk assessment models using cells for the study of endocrine disrupting chemicals, and in my case, PBDs. So, two of my main objectives were therefore to characterize the mechanism of toxicity of a PBD mixture with my cellular model, and to evaluate its effect on cancer progression using thermosphere or mammosphere in 3D. And of course, we kept in mind the results obtained in vivo to evaluate their relevance. For my project, I worked with two luminal cell lines of the mammary gland epithelium found lining the walls of the lactociferous ducts and acini. These grape-like structures are connected to the nipples, forming a functional mammary gland capable of ejecting milk for the infant. I've worked with the MCF12A, a non-tumorigenic cell line, allowing us to evaluate the effect of our compounds in a non-cancerous context, as well as the T47D, a tumorigenic cell line expressing hormone receptors associated with breast cancer, namely those for estrogen and progesterone. And because of time today, I'll be presenting results for the T47D. Now, this would be a good time to introduce breast cancer a little. So this cancer is the most frequent cancer developed in Canadian women, with 1 in 8 being diagnosed. This disease develops in different stages: initiation into unaffected cells, promotion, which is characterized by anarchic proliferation of cells, and progression, during which the benign tumor becomes malignant thanks to mesenchymal characteristics, such as an increased ability to migrate in neighboring tissue. Now, the sensitivity of the mammary gland to hormones plays a crucial role in orchestrating its development during puberty and its remodeling during and after pregnancy. However, it is also this characteristic that makes it susceptible for endocrine disruption. And this is why exposure to endocrine disruptors is increasingly becoming a potential risk factor for the progression of breast cancer. So the exposure mixture in my project differs slightly from the one in previous studies with rats of our laboratory. The former consisted of PBDs, including 5, 7, and 10 bromine congeners, as well as HBCDD. And it was representative of the concentrations found in house dust and was administered to rats orally, to the dams. My mixture, on the other hand, is representative of the concentrations of PBDs in breast milk and contains only PBDs. So, the first test we conducted aimed to measure cell proliferation in real time using the XLHS. In short, the testers measure the cell adhesion to the bottom of the wells, and this measurement increases with proliferation. These data are obtained every 10 minutes, hence the real-time nature. This technology allowed us to test up to 8 concentrations of our mixture at the same time, and our median concentration, 38.6, represents the average value detected in breast milk. As you can see, only the highest doses caused an effect on the cell proliferation and caused a decrease. Although toxicity was not specifically tested, we could Sorry. I received the notification that I was muted. Everybody's hearing me well?
Yes, we hear you well now.
Okay. So for further experiment, we did not include these concentrations. Except for this second experiment, where we also used the acceleration instrument to study the cell migration. This was achieved using a tool that can replicate the scratch test principle in all the wells of the microplate at the same time. So once the confluence is reached, the scratch is made, causing a cell loss, and the signal drops to 0. And then cell migration in the vacated space is measured in real time. So once again, only the highest dose affected the migratory capacity of our cells. And you may also notice there was a lot of variation in our data, which we believe it could be explained by the fact that actually T47D cells are not known to be highly migratory. Now, to understand the toxicity mechanism of our mixture, we also looked at its effect on the expression of certain protein of interest in breast cancer, and we used the Western blot technique to quantify the expression of hormone receptors involved in breast cancer. So first we have the estrogen receptors. Estrogen has a role in mammary gland development and allows for the elongation of the lactociferous ducts, which depends on the activation of both receptors, ER alpha and ER beta. Our results show a overexpression of both receptors in the presence of our mixture in the T47D cells. This disruption of the normal expression could, for instance, suggest an alteration in its sensitivity to therapy. It has also been shown that the activation of ER alpha can have pro-tumorigenic effect on cell proliferation. Then we have the progesterone receptors, because progesterone is another hormone implicated in the mammary gland development, and its deregulation can also be linked to breast cancer. Our results show a significant decrease in the progesterone receptor expression in the RT47D cells. It is known that the progesterone pathway can crosstalk with the ER pathway, and therefore the decreased expression here could be linked to the ER overexpression, since the hyperactivation of this crosstalk can downregulate PR. And finally, we have the thyroid hormone receptor. This receptor plays an important role also in regulating the mammary gland development and lactation. And BFR, especially PBDEs, have often been shown to disrupt thyroid function, including in the mammary gland. The exposure to PBDs has also been linked to thyroid diseases in humans. And although our statistical test did not reveal a significant difference with the control, we can see that the expression of this receptor tends to increase in the cells. Now, the second part of my project focused on developing a 3D cell model for studying the toxicity of endocrine disrupting chemicals. This model, called spheroids or tumospheres in the case of cancer, is more representative of cell behaviors in vivo compared to cell culture in a model area in 2D. We use these models to evaluate the effect of our mixture on growth and on the tumosphere viability. There are 4 main steps in the formation of our spheroids. First, cells are seeded into a non-adhesive round bottom microplate, followed by rotational agitation. And next, we had an extracellular matrix product to provide cells with the structural proteins needed to form the spheres. And then, again, rotational agitation. Now, cell growth of our spheroid was measured by imaging after 4 days of exposure to our mixture. And as you can see, we did not observe any difference in the tumor growth. And in a way, this coincides with the proliferation results we obtained in 2D. We then studied the effect of our mixture on the metabolic activity of our tumospheres using resazurin salt test, which is a molecule that is reduced into resorphin through mitochondrial activity. We observed here a decrease in the metabolic activity in our spheroids exposed to our mixture, and such a decrease can indicate an effect on mitochondrial integrity, proliferation, or that the oxygen and nutrient gradient conditions are worsened by the mixture. And finally, in order to compare our 2D and 3D results, we also studied the effect on the markers expression. So here we can see that the estrogen receptors were not as significantly affected by the exposure, contrary to our results in 2D. For the progesterone receptors, we can see the downward trend that is similar to our results in 2D. These results reinforces the hypothesis that progesterone receptors are affected by PBDEs. And finally, we again observed an increase in the expression of the thyroid hormone receptor. Now, this result initially surprised us as it seemed to contradict the metabolic activity findings. However, we've seen that it has been shown that this receptor, when on-ligand to the hormones, acts actually as a transcriptional receptor, a repressor of thyroid hormone responsive genes, including those implicated in the mitochondrial activity. This could therefore explain the link between these two results. So what should we remember? Well, first, we observed no effect of our mixture on the proliferation and migration in 2D and no effect on the growth of our 3D models. However, in 3D, the mixture disrupted cell metabolism. And finally, both in 2D and 3D, the mixture seems to affect the hormone receptors associated with breast cancer. And interestingly, previous in vivo results have shown disruption of these receptors as well. So I am very proud of this project because it has revealed new data on the toxicity of PBDEs in an environmentally relevant mixture. We have developed a 3D cellular model for studying endocrine disrupting chemicals in breast cancer. And finally, some of our results could even lead to the identification of important signaling pathways in breast cancer. So, of course, I'd like to thank my director, Isabelle Plante, and all the members of her lab for their support and technical assistance. I also thank Mashid Babdad and the members of her lab for the assistance on developing the 3D model. I thank, of course, all the funding organization. And before I finish, I wanted to have a small talk on me being a part of the CIAPE, which brought a lot to my PhD years. I've been part in the training committee, and we've worked on many different projects, such as the podcast, the journal. We did some popularizing nights at the pub, which was hormones and hops. We also participated in the contest Défi Oser Entreprendre, and we won some prizes. And of course, being part of the CIAPE gave me confidence to do presentation abroad, which I'm very proud of. So thank you also to the CIAPE, and thank you to all of you for being here today.
Thank you very much, Madeleine. That was very interesting. I'd like to open the floor to questions. Any questions from the audience yet?
No, we don't have any question yet.
But, well, then I always have questions. I was intrigued by your link to thyroid hormones and thyroid hormone receptors. Are thyroid hormones typically associated with breast development? I mean, I've always, been aware of the estrogen receptor and the progesterone receptor, but can you tell me a little bit of the background on the thyroid involvement, please?
Yeah, so it is true that we talk more about progesterone and estrogen, but the thyroid hormones are like indirect regulators. So their regulation could eventually be linked to breast cancer in a way. They're more into the regulation of the remodeling and lactation, if I'm not mistaken. But I'm starting to read on that to create some interesting discussion on those results.
Oh, well, thanks. I mean, that was an interesting connection, and one that, as an outsider who does not study aspects of breast cancer, that I wasn't expecting you were going to go there.
Mm-hmm.
So that was very interesting.
Thank you.
Did that give some time for people in the audience to come up with some questions? Great.
We have another question in the audience. Bernard asks, what do you think, Madeleine, is the basis for the different results between your second and third studies?
I think he meant 2D and 3D models.
Yes, 2D and 3D studies. Sorry. Yes.
So the 3D models forms a structure where there is gradient of, oxygen and nutrient penetration, but also for the compounds penetration. So I believe in a 3D spheroid or tumosphere, the compounds does not reach every cells in the same way that it does in a 2D layer. So it, in a way, represents more of what happens in vivo in this format. But on the other side, in the epithelial of the mammary gland ducts and acines, we have a wall of cells. So I think both models are important to consider in the breast cancer context.
Any additional questions?
No, not yet.
Okay.
Q&A and answer later on.
Yes. Thanks. I also found it interesting that you spoke at the very end with the little snippets about how your involvement with the network had, promoted your development. And
yes.
I appreciate you taking that time to to make those comments. Sometimes perhaps we tend to overlook both the social aspects of that in creating a community, but also in sustaining that community. So thanks for that gentle reminder.
Yes, I would recommend every student to try to get involved in the CIAPE. It was really a plus value in my PhD years.
Well, great. Well, thank you very much, Madeleine.
Thank you, everyone.
Yeah. Well, maybe we can continue to our third speaker. And